in a very informal manner. So if there's anything that you want to ask, you can just unmute your microphone. I will also ask you to show me a thumbs up every so often, so I can know that things that we're trying to do, you actually were successful in doing. I would assume that everybody has by now a gEAR account, and also hopefully you signed in the sign-in sheet. So we're going to skip setting up your account, and we're going to start straight from the compare tool. So the compare tool is designed to quickly appreciate differential gene expression across any two conditions. And as a disclosure, the compare tool is one of my favorite tools in the gEAR, because oftentimes when I'm looking at the data set, all I really want to know is, which genes are higher or lower in one condition or another. It can be between cell types, or someone published a paper about a mutant I really care about. And what I really want to know is which genes are up or down, and get access to that. And then I also might want to know my genes. So it might be our RFX3 or another gene. I want to know how does that gene, my gene, behave, and my gene might not have been something that they put in the tables in their manuscript. So we're going to demonstrate that. We're going to go to the gEAR main page, and we're going to choose the hair cell profile. Just put a gene name in, like PAL4F3, that we all know. And for those of you who are not aware, there is an exact match button here. So we'll press on exact match. If you want to see things that are not exact match, then you don't press on it. For example, if you're looking at a gene family like socks, you might want to see all the soxes. But right now we'll do exact match, PAL4F3, and the hair cell profile, and press on search. And we'll give it a second, and it will call all of the data sets that are focused on hair cells that we put in this profile. You can see at the top we have our functional annotation that if we click on it, it will give us more information in a species-specific manner. If we unclick it, it will disappear. PAL4F3 is a deafness gene, both in mouse and human. So these buttons highlight, and if you click on them, they will take you to the information. But what we really want to do is we want to find the data set by David He, and that is called RNA-seq, Dieter cells, pillar cells, inner hair cells, and outer hair cells. So we're looking for this specific data set. Can everybody find this data set in their page?
Yes.
Right, and we're going to click on the inverted triangle, and we're going to click on the compare. We'll open a new tab. All right, so let me know when you got to the new tab.
Got it.
Got it? All right, terrific. So, so now we're all going to get very curious about comparing gene expression between inner and outer hair cells. So the top here tells us that we're looking at the data set that we chose. We're going to choose inner hair cells, and we're going to then ask to compare it to outer hair cells. It tells us that the data here are going to be log2 transformed. This is appropriate for this data set, but for some of the analyzed single cell data sets, you might not want that. It also tells us that it's only going to show us genes that are at least twofold different between the two conditions. This is important to understand, because there are sometimes data sets that don't have any genes that are twofold differentially expressed, or maybe sometimes there are two conditions in a data set that are very similar, and you might not see many dots or any dots at all. So if that happens, you might want to change this to 1.0, but here we're going to stick with 2. and you're going to press on plot. This is working, and once it's done working, you should all see something that looks like that. Please let me know with a thumbs up when you can actually see a dot plot as shown in my slide. Okay, terrific. I see that too. All right, wonderful. So we're seeing a dot plot that shows differential expression between inner hair cells and outer hair cells in this particular data set from the lab of Dr. David He. The middle part that is whited out are genes that are less than twofold differentially expressed, but we're seeing dots and we might not know what they are. Let's say I'm very curious about genes expressed highly in inner hair cells. If I hover over any of these dots, it will actually tell me what's the gene name. So you should be able to do that yourself. But the problem is that what if some of these genes are differentially expressed, but they're not statistically significant? Now, this is not instead of having a bioinformatician or running official tests, but if you want to get, just a quick idea, then you can choose any of the statistical tests that are here. For example, choose a non-parametric test, and we'll plot again. And now the genes that are statistically significant according to the significance test that we chose and the p-value cutoff that we chose are going to be colored red. Please a thumbs up when you see that on your screen. Dylan, can you get that to work? Yeah, all right, terrific. Okay, so this is a lot of dots and they're both red and gray. So if you want to see, for example, only the ones that passed the cutoff, you can click here on did not pass the cutoff. And now all the ones that did not pass the cutoff will disappear, or you can make them reappear like this. Okay, and now, however, you might be very, very interested in knowing where some of your favorite genes are expressed. So if you type here, let's type ATP to A3, which is an inner hair cell gene, and maybe SLC 26A8. I think that's it, right? No, SLC 17A8. Or any other gene, and we'll press plot. What you should see in a second is that the genes that you typed in are going to have arrows associated with them, okay? And if you happen to plot in right now also another gene that you really like, here you can see the genes, the ATP2A3 has an arrow and the SLC has an arrow. It's red and red, so it's hard to see. But if you happen to type in another gene that you really like and you don't see it, it is very possible that it's not differentially expressed, and that's why its dot is not appearing in the middle here. But oftentimes what we really want to do when we look at differential expression across two conditions is we want to generate a short list that we can work with. So what we're going to learn now is how to generate that list. there is a lasso tool that allows you to select any group of genes. We're going to try to select a small number, so it's going to work fast in the workshop, and that will generate a table. So I'm going to choose the lasso tool here, and I'm going to mark a small group of genes that are high in inner hair cells and are low in outer hair cells. And once I close the loop, there's going to be a table that appears on the right with the fold change and the p-value according to the test that you chose, which may be the most appropriate one or not. And this table you should be able to download by clicking on the download link, and it will download as a text file. Then give everybody a minute to select a group of genes and generate the table, and hopefully see the genes that they searched for earlier highlighted in that table. Okay, great. See, Royce has it. Bazil, you got it? Good. So the table we can download, and it's very useful in the very near future. You're also going to be able to build your own table with your own lists of favorite genes. So let's say you're very interested in genes of planar cell polarity, and you always want to look at that group of genes. You'll be able to build that cart and always have that available to you. But right now, the carts are built based on differential expression in the compare tool. And you can click on the cart and give it a name, like Inner Hair Cell Workshop 2. Give it a meaningful name and save it. When you do that, this cart is now saved into your personal account. And if we go back to the gEAR, and we go back to the, we'll go back to hair cell profile. We're going, actually, why not just do the main profile, the hearing profile. We'll press exact match just with something simple. So file 4.3. But now that we're all back in the main profile, instead of gene, you'll see under the profile in the upper left, there's something that says gene cart. It's a very small print. It says the gene cart is empty. But if we go ahead and we click on it, Workshop 2, or whichever gene cart you named it, the list that you generated will automatically populate the search tool. And if you press on search, it will now run a search for these genes for differential expression in the profile that you selected. So it's going to take a minute because it's a long list of genes. Okay. And here you see, for example, the top gene that shows it's just organized by alphabetical order. So ATP2A3 it came up as high in inner hair cells in the data set of Dr. David He. And you can see here it's also expressed in a very small number of cells that happen to be inner hair cells. Now, look at this. Here it's expressed in what looks like all hair cells. But that's why you need to read the title, and you need to understand the data set. This data set took all of the hair cells in the cochlea and all of the hair cells in the vestibule separately. And so the signal is really coming, now you know, that's why it's so useful to have multiple data sets in one page. The signal is coming really from the inner hair cells, but it's coloring here all of the hair cells because this data set, it's pooled inner and outer hair cells together. If we go to the next, another gene here, I think it's another cool example. So DNA JC5B, you can see it's expressed in inner hair cells and P1 in the single cell from Dr. Kelly, a little bit in the outers. In our data set at P0, we didn't detect expression actually in a meaningful manner in hair cells. It makes sense, there's very low expression, but we saw really high expression for this gene in vestibular hair cells. And you can see that this gene is really highly expressed in zebrafish in a cluster. And when you look here, you see that it's really highly expressed in the cluster of hair cells in zebrafish. And the gene also has a diurnal variation in expression, according to the Canlon data set. So you can see that by putting your favorite genes and now scrolling at different data sets, you get a lot of information. Okay, so what we're going to do now is we're going to jump from here to the workbench. Okay, so we learned how to do really simple comparisons of two different conditions and the regular data sets. This, by the way, works also for the single cell data sets. But now we might want to look at some single cell data sets in a more meaningful way. And so we're going to put in KCNQ4. And we're going to choose up top here the lateral wall profile. Please show me with a thumbs up once you are in the lateral wall with KCNQ4. KCN. So this profile has, okay, great. Has many of the lateral wall data sets that were presented yesterday in the gene regulation session from the laboratory of Michael Hoa at the NIDCD. And we're going to navigate all the way down to their most bottom data set called adult mouse single nucleus RNA-seq methanol fixed from the Dr. Hoa lab. And you can see here that KCNQ4 appears to be expressed in two of their clusters in the intermediate cells, and what is this one? Blue in the basal cells. But you might want to know, for example, let's say you have your own data set that you generated and there are a number of clusters there, and you want to find out what are the best marker genes, right, for each one of these clusters. And you don't want to sit here and start to guess. So to do that, you will click on the inverted triangle and press on Analyze. And when you do that, it will open this data set in our workbench. And you'll be able to do two things here. One thing you could do is you can actually do a de novo analysis. You can analyze this data set from scratch, and I can tell you that you will get to something very similar to what they got. And if you didn't get a chance to go to breakout room 3, then the handout for breakout room 3 is available in the workshop website, and you can learn how to do that. And we'll also put the videos there. But you know, much faster and more useful maybe is to just go to the primary analysis. So in the stored analysis, we're going to press on primary, and then you can just type in how many marker genes you might want. So let's look for the top 5 marker genes for each cell type, and we'll press on go. Please let me know once you were successful in getting this to work. All right, I'm seeing some thumbs up. And so the marker genes will appear in 2 formats. One is in these tables that you can right click and open in another link in another tab and zoom in. The y-axis shows you the strength of the marker gene, but more useful in my opinion is below here we have a table. And the table tells you the top marker genes for each one of these cell types. You can download it, but you can also just click on any one of these marker genes. I will now click just on the top for every single cell type. And you can also type in additional genes, for example, KCNQ4, and press on visualize. And what this will do, it will generate a dot plot for you that will allow you to immediately see gene expression across multiple cell types. So you can see here, for example, that this gene here is a really great marker for fibrocytes. Or just this gene here is a really good marker for marginal cells, but it's going to have still some expression in their cluster they called unknown, and maybe a little bit in the Reisners. So you get an idea of gene expression across clusters, and you can add whichever number of genes you like to this map. You can right-click it and see it larger. You can download it. But if you're really interested in comparing between any two clusters, then you're going to press on the compare gene or clusters, and you're going to scroll up. And now you can choose to compare between any two clusters. So for example, between marginal cells and let's say the spindle cells that we learned about only yesterday, you choose your statistical test, you choose how many genes you want, and press on go. And this is extremely, extremely helpful in my opinion, because it really gives you the most granular information about the differential expression, because every single cell in the data set here will be represented with a dot. And so you can really find your very best markers to differentiate between any two cell types very quickly, very easily, without having to download the data set. So here you're going to see on the top genes that are higher in marginal cells compared with spindle cells, and here genes that are higher in spindle cells compared with marginal cells. And you can show a table first that shows you also the statistical significance, or download it. But if we just write down, say, of markers, for example, CACNB2, and now go back to the page we were at before and see whether this really works in other data sets, CACNB2. It was supposed to be a marker for marginal cells. And you can see here that at the top data set, it was expressed in this cluster that is marginal cells. Then in another data set from Dr. Hoa's lab, it was expressed in this cluster that is marginal cells. And another data set was expressed in this cluster that's marginal cells. And here in another data set, it was expressed in this cluster that's marginal cells. And again, so you can see how very quickly from the compare clusters tools, you can get very meaningful information about identifying differentially expressed across clusters, and do in silico validation, really, right? I mean, you see it validate to this extent, and in 5, 6, 7 other data sets, you don't really need to do in situ hybridization to confirm this result before you do something with it. And a lot of people cite the gEAR in particular data sets in papers or grants to support their hypothesis. So this is really what I wanted to accomplish in this room. I can take questions, or we can jump back to the main hall. All right, so let's jump back. Oh, go ahead.
I have a small question. We generated in our card a file that we saved, something like workshop. Is it possible to delete it, or would it remain there like forever in our accounts? So
that's a great question.
Remember in the first half.
Yeah. That's a great question. So right now, we can't edit the gene cards, but we're in the process of building a new data set manager that will also allow you to manage your carts. So I hope that within the next couple of months, that functionality will be available to you. But right now, the gene card list just gets longer and longer, and that's a deficiency that we are working to correct.
Okay, thank you.
Also, anytime you have a question like this, a feature that you'd like, you're doing something for the gEAR and you're just thinking, I wish the gEAR would be able to do this for me. There is this little button here. It says ask us questions or give some feedback. When you press on it, just fill out this form, and we want to hear from you. We want to improve the tool so it will serve you best. All right, I'm gonna stop sharing and let's jump back to the main room.